Good evening everyone. This is your week 10 session. So in this session we'll be talking about week 10 topics which were related to molecular dynamic simulations, in which you have looked into that what is MD simulation, why do we need it, and you have looked into the detail of the simulation process and what is the requirement of each step, that also you have looked into. Then you have also seen the concept of force fields, energy minimization, different methods. So we'll one by one look into each of these concepts, and then we'll practice few MCQs. Along with these topics, we'll also see what is the current literature scope, and I'll take your doubts. So let's begin with the session. So first of all, we'll look into what is molecular dynamic simulations. So when we say molecular dynamic simulation, it is a computational technique which we generally use to analyze the motions and interactions of atoms and molecules. For example, in this figure you can see that these circles, they are different atoms which are connected by these bonds, and now they are interacting, they are moving in the system. So these interactions, their movement, so this dynamic study which we are studying using the computational methods is known as molecular dynamic simulation. So how it works? So it uses the Newton's equations of motions to determine the movement of particles, and it also applies certain mathematical models to predict the molecular behavior. So according to Newton's equations, it also says that a particle which is in motion, so there is some kind of forces which are acting. So each particle is moving with certain velocity, it has some mass. So all those equations come into picture when we study the dynamics of a protein. So now why it is important to study molecular dynamic simulation? So first of all, it provides us detailed insight into the molecular interactions at atomic levels. So at the level of atoms, we can see that how the movement is happening, because we all know that protein is getting folded into certain tertiary structure. So that folding is happening in which different atoms are coming closer to each other. So they are taking some conformations. So there is something which is happening at the level of atoms. So we can study those interactions with the help of MD simulations, and it also helps us to understand the structural and dynamic properties of biomolecules. So with this we can see what is the structure of a protein, and dynamical means with time how the structural things are getting changed, whether it is getting folded or unfolded. So if we study the dynamical properties of a protein, then later on we can comment on its functional properties as well, that at particular temperature, for example, a protein is getting unfolded. So in that way we can study those things using molecular dynamic simulations. So applications are protein folding, stability analysis, drug discovery. So we can also design certain drugs, and we can see that how a particular drug changes the conformation of a protein, because indirectly it will influence the protein function. So all those studies can be done. Protein ligand interactions can be studied, enzyme mechanisms can be studied, that how a particular enzyme binding to substrate, how it initiates a reaction and how it catalyzes it. So what are the conformational changes that is happening and how product is getting synthesized, so that we can make some modifications, we can design certain other substrates, or we can change the reaction process. So all those things can be done. Then nanotechnology and material science, so they also uses this molecular dynamic simulations, and few of the MD softwares are Gromax, Amber, NAMD and Charm. So in this case also, this is also one of the representation which shows that how the particle is moving across the membrane. So these are some of the transmembrane helices which are spanning the our membrane. So this is showing how the movement is happening. So in this scenario there is different means how the atoms are moving. So inside this helices are moving, how this particular substrate is interacting at different sites. So all those studies can be done with the help of MD simulations. So this is also another slide which talks about that why do we need MD simulations. So because we have certain limitations of experimental methods, because experimental methods are costly and they are also quite complex, and there are certain conditions which are difficult or impossible to create in a lab. For example, if you want to study galaxies in the stars, what is the conditions, so all those things cannot be recreated, or it's quite difficult to study them directly. So what we can do, we can use the simulations to study them. So if I give example from the real world, then you have also seen that for example there are certain to study that how during the crash airplane crash, how the conditions have varied, what was the scenario. So all those things cannot be recreated by performing the original scenario. So what we do is we simulate those conditions and we see that how the machines behave in those conditions. So that is the example, that what are the limitations at the level of experiments. So for which we need MD simulations. And then observing atomic level interactions in real time, it is also quite challenging. So how MD simulation helps in this? So we can simulate molecular behavior under different physical and chemical conditions. So these conditions the control is in our hand. So we can alter these conditions and we can see that how molecule is behaving differently in different conditions. And if we need to perform it in the wet lab conditions, then it's quite complex and it will take a lot of resources. So that's why MD is better than the wet lab experiments. Then provides data on structural stability, dynamics and reaction mechanisms. And then it can also be used to predict the experimental results before actual lab testing. So although we all know that lab testing will give us the result which will be 100% confident, because those situations are really happening. But the advantage of MD is that we can figure out, or we can understand that whether this result will give us, means we can get some idea that whether we should proceed with this experiment or not. So in that way it can help us to shortlist certain proteins or shortlist certain criteria, so that we can proceed with those instead of performing the ensemble of calculations or ensemble of experiments. So this can help us in shortlisting. And key advantages are that this is safe, because we do not have to deal with certain chemicals or radioactive or temperature or pressure conditions. So that's why it's safe. It is cost effective, and it also allows us to perform in-depth analysis of molecules. It enables the study of biological molecules in their native environment. So with these simulations, we can also make changes on the basis of the native environment. We can add water molecules or all those things. So that is possible in these simulations as well. So that's why we can study the protein in its native environment. And this is also one of the representation that there are multiple things which we can study. We can perform all atom simulations, or we can also perform coarse grained simulations. So I will not go into their detail. In the later slides we'll see what are these things. So we can understand the secondary structure content. We can also see what are the interactions that is taking place. So in this way, if we conjugate or if we use multiple simulation techniques, then we can improve different analysis such as thermal stability, bioavailability, solubility, bioactivity, enzyme catalysis. So all these properties can be studied with the help of different MD simulations if we combine different results. So this is one of the paper which is still up in bioarchive. So it's a preprint, it's not published yet. So which talks about the design of facilitated dissociation which enables the control over cytokine signaling duration. So why this paper is important? Because it tells you how the property or how the protein molecule changes as soon as the cytokine mimic binds to the protein. So there is some kind of dissociation and structural changes which are taking place. So this study is possible with the help of MD simulations. So with the help of simulations, we can understand that what are the changes taking place in the real scenarios. So this research is crucial because it provides a novel method to control protein-protein interactions dynamically. So in this way, they are designing different cytokine mimics, and they are checking that which mimic will lead to this dissociation of this complex, which is the common gamma chain of this particular protein. So they are studying this particular, how the interactions are taking place. So they are designing different cytokine mimics. So if we have to proceed it using the experimental methods, then it will take a lot of time, and it will also take a lot of resources as well. So in this case, this MD simulations can help you to shortlist certain cytokine mimics which are giving you better results. So the ability to rapidly dissociate protein complexes such as cytokine receptor interactions has significant implications for therapeutic designs. So it's involved in some therapeutic purposes which allows for the more precise and time sensitive biological responses. So now we'll see that how MD is playing their role in this study. So MD reveals excited state transitions. So it helps model and predict how design protein transition between stable and strained excited states, which enables the controlled dissociation. So it will tell you that what is the protein coordinates before binding to cytokine and what are the coordinates after binding to this cytokine mimic. Then we can also understand structural frustrations. So which means it gives you the information about the structural strain and energy landscape. So it is also possible that as soon as it binds, there are some helices, they might come closer, and there can be a straining which is happening on different molecules. So due to different strains, the protein might get destabilized. So we can also study the energy landscape, that either it is in stable conformation, or it is unstable. So that study can be done with the help of MD. Then validating design hypothesis. So by simulating interactions at atomic level, MD simulations allowed the researchers to test and refine their induced fit dissociation mechanism before experimental validation. So as I was telling, that instead of using all the cytokine mimics and then testing them in experimental conditions, we can shortlist some of the cytokine mimics which gives better results in this MD simulations. So overall, MD simulation can help as a guide to tell you that which of these mimics can give better results, so that we can experimentally test them and later on confirm our hypothesis. So this is about this. And if we talk about the detail of this figure, so it shows you the artificial superagonist NEO2, which is this cytokine mimic. So it enables rapid and controlled interleukin 2 signaling shutdown. So in normal cases, interleukin 2 signaling happens if there is some kind of a viral encounter or if there is some infection in our body. So interleukin get activated and they start a series of reactions. So it will create a trouble if you have some condition of autoimmune disorder. So in that condition, interleukin 2 get upregulated and it interfere with the normal machinery. So we want to suppress the signaling. So in that case, we need a cytokine which could bind to this and leads to dissociation of this common gamma chain. So this mechanism shows you that how the dissociation is happening, and this is known as induced fit facilitated dissociation mechanism. So certain conformation of this mimic is responsible for properly binding to the protein, interleukin 2 protein. So this is about the paper. Now we'll look into the history of MD simulation, that how it began. So early foundation was in 1900s. So introduction of force field concept in molecular interactions. So whenever we study these interactions, so we'll see in the later slides that the force field concept is very important, because there is some interaction happening between the atoms. So that could be stretching, bending, rotation. So all those things are taking place, as you can see here as well. So between different atoms, these are the different types of forces which are acting, which will make or which will bring about the change in the position of atoms, or you can say which changes how the molecules can interact. So all those things come under force field. Force field will tell you what are the different measures which you can consider while studying the protein dynamics. Yeah, so there are different types of interactions have been shown, such as torsion angles, bond length, bond angle, then Lennard-Jones potential. So there are some interactions which happens due to the binding between the two atoms. So those interactions are known as bonding interactions or bonded forces. Whereas some interactions can also take place where the atoms are not bonded directly with a bond. So these are some far range interactions. So these are known as non-bonded interactions, such as Lennard-Jones potential. So these are van der Waals interactions or non-covalent interactions. So they comes under the non-bonded interactions. Now in 1929 to 1953, it was a developmental phase for these MD studies, in which atomic potentials were formulated. Monte Carlo simulations were also introduced for statistical sampling. Then in 1957 to 77, first MD simulations came up. So there were two scientists, Elder and Wainwright. So they performed MD on hard spheres in 1957, and then in 1977, first protein folding simulation was given by Levitt and Warshall. Later on in 1980s and 2000s, it was a growth in computational power when multiple computational resources came up. So it kind of helped us to enhance the knowledge of MD simulations. So there was development of specialized algorithms, force fields such as charm, Ember, Gromos. So these are the examples of different force fields. So they were developed, and it also improved the accuracy of biological system simulations. And if we talk about modern MD, which is after 2000 till now. So that involves high performance computing, GPU acceleration, and different AI integration to make sure that which force fields to consider, what weightage should be given, what should be the threshold. So all these developments have taken place till now. And these are some of the names of force fields, such as amber, or it was also known as OPLS. So it is a type of all atom or united atom force field. And these are some of the references. So if you are interested to read in depth about these different force fields, then you can go through these papers. This tells you that this force field is applied to all atom, or for example in this case rigid valence geometry. So these are some types of force fields. It tells you that on which atoms it is acting on. This is also another perspective, that how MD simulations have developed through time. And in this, different scientists' name has been mentioned and what work they have contributed towards the development of MD simulations. Now I have found another paper which was about the development of molecular dynamics and research progress in the study of slag. So in this, they have very clearly mentioned that what are the different steps in MD simulation. So first of all, we need to build a model. Then we give the initial conditions and preparatory work. Then calculations and solutions are given. Then data output and correlation. Then we get radial distribution functions, logarithmic distribution functions. So in later slides, we'll one by one see in depth about these different steps. So what else that tells you that this molecular dynamics, it can be applied to different fields, such as nuclear science and technology, so in which single crystal tungsten, nuclear research, radioactive waste. So all these studies can be performed with the help of MD simulations, and this can also be applied towards radioelectronics, in which we can study the dislocation nucleation, nano oscillator, phase transition mechanisms. Then power industry, then metallurgic engineering, mechanics, oil and gas, geophysics, hydrodynamics. So these are different fields in which molecular dynamics can be applied other than the biological field. So these simulations are quite advantageous. Now if we talk about the MD simulations in biological systems, now we'll see that in where we can apply the MD simulations. So first of all, why MD simulation is crucial in biology. So if we talk about different biomolecules which constitutes our body, so they are proteins, DNA, lipids, so they constantly change their shape. They constantly undergoing different interactions, different processes, and bringing about the effect at the level of phenotype. So MD helps to track these changes, that how interactions are happening, what are the changes that are taking place in different conditions. So we can compare among normal and disease situations, that what are the interactions which are getting changed. So with the help of MD simulations, we can track those changes. So it helps the model molecular interactions, such as protein ligand binding, enzyme activity, or membrane dynamics. So in this figure, I have shown that how a protein is getting folded. So towards the x-axis, this is the time, that in this time period, how the protein is changing its conformation and how it is getting ultimately folded into its native state conformation. So here it was unfolded. Then different helices are aligning together, they are coming together, they are getting folded. So key application is protein folding. So we can understand this protein folding because in MD simulation, we'll see that with time, how the molecule is changing its dynamics, how it is changing its positions, its coordinates. So that way we can understand the protein folding mechanism. It also helps us in drug designing. We can also identify different drugs and how they are binding to different protein targets. And after binding to targets, whether they are bringing out some change in the conformation. So all those studies can be done. Then membrane simulations. So we can also study the transport mechanism, that how a particular molecule is getting transported through the membrane, where it is getting interacted. So all those things can be studied with the help of this. Then nucleic acids. So we can also investigate the stability and interaction of molecules and nucleic acids. So experimental correlation, MD helps to interpret the X-ray crystallography and NMR spectroscopy data. So this interpretation can also be done. So as in this case, I have shown you the protein. For example, if it is a drug, then we can see that in which conformation, or if we change certain constituents, certain residues, then we can study that how the interaction is changing, whether it is bringing about change in the protein conformation, Or we can also see which of the docking pattern of this particular drug is best, so that we can change certain dihedral angles, or we can change the geometry of this particular protein. Yeah, so this is also that how we can simulate the nucleic acids, and how we can study that how the different changes are taking place with time by altering certain conditions and all those things. Yeah, so this is an overview that what a molecular dynamic process is composed of. What are the different steps which are involved? So first step is system preparation. So in system preparation, firstly, we need to define the molecular structure. We define whether we are taking a protein or it is a ligand or a solvent. So we define all these things, we provide all these things. Then we assign the atomic charges and connectivity. So we know that a protein can be charged. It can have some positively charged amino acids, negatively charged amino acids. So those amino acids can bring about the overall charge in protein. So we can assign those charges. Then comes simulation setup, in which we apply different force fields. For example, charm, amber, chromos. We set temperature, pressure and other environmental factors to study that if we want to study a protein in particular temperature conditions, so we can study them by defining the temperature, by defining constant temperature conditions, or we can set a constant pressure, we can change it, so we can study the fluctuations with respect to changing pressures or temperature conditions. So all those things comes under simulation setup. So this is our second step. In the second step, along with this, we also add hydrogens. We generate a topology in which a particular protein's covalent structure is mentioned. Then we build a simulation box. So simulation box defines that which part of the protein you want to focus on. So in that case, you will define the simulation box. Generally, that box, if a protein is very large and you are only interested in the active site, if you want to study binding of a ligand at the active site, so your simulation box will comprise of the near active site residues. It will not focus on entire protein. So it kind of helps you to study the significant or specific conformational changes of particular protein. Then come additional atoms. So in case if you have a charged protein, then to neutralize it, you can add certain ions to neutralize the charge and then later on proceed with the simulation. Then our third step is simulation execution. So till now you have defined different conditions. So in execution, you have to equilibrate the system, in which you stabilize the system so that it is in a stable conformational form. Then you perform production run, in which you collect the molecular motion data, that how it is changing with time. So in molecular dynamics, the main purpose is to study that with time how my protein conformation is changing and which of the conformation is most stable or is the native state conformation. So that thing we can study. So in this case, in production run, we will collect this molecular motion data, that how the dynamic is changing. After we collect this data, the next step is post simulation analysis. Now you have performed the analysis of with time points, so now you'll analyze it. So for analysis, what we do is we perform RMSD calculations. So RMSD stands for root mean square deviation, in which we have a wild type structure and then we have the structure which we obtained from the MD simulation at different time points. So we compare it with our wild type structure, that how the conformation is changing at each time point. So that the difference and the position or in the coordinate of each atom, that is known as RMSD, that how much deviation is present between the native structure and the structure which we are observing. Yeah, so this also we'll cover in the later slide. What is the exact formula? All those things will be cleared. Then comes energy profiles. So we can compare the energy profiles, that whether my protein is getting destabilized. So in that scenario, its energy will be higher. Or if its energy is low, then it means it is a stable protein. So those comparisons can be done. Then interaction analysis, we can also analyze that whether some new interactions are getting formed, or whether my protein, if my protein is converting from unfolded to folded state, then there is a possibility that different hydrogen bonding is taking place to bring about the proper protein folding. So in that scenario, we can perform the interaction analysis. Then visualize trajectories to interpret molecular behavior. So with this, we can study that how my protein is getting folded, how many different transition states it is passing through to get into its native conformation. So all these analysis can be given by the molecular dynamic studies. Yeah, so this is also the example that how my protein is present and these are the water molecules, so that I can study the protein in its native state where it is present in the aqueous environment. So we study different parameters. We study the positions of each residue or each atom, you can say. We study the velocity, that with time how its position is getting changed. We can study the acceleration, we can study force which is acting. So overall force we can study the potential energy, that what is the potential energy of my protein, whether it is high, it is low. So this energy will tell you that whether your protein is stable or it is unstable. Yeah, so the main thing which comes now is the force fields, because the potential energy can be calculated with the help of force fields in MD simulations. So force field is a set of equations and parameters which describe the atomic interactions. So how we can study that what are the interactions taking place? We define the force fields. So they consist of a potential energy function. That means it involves both the bonded and non-bonded interactions which are happening inside a protein. If we study those interactions, then only we'll be able to see that what is the net energy is on the system. And a parameter set which is the numerical values for bond lengths, angles, charges, etc. So if you want to calculate total potential energy of a protein, then it is composed of bonded plus non-bonded terms. So in bonded interactions, that is energy of stretching, bending, torsion, and in non-bonded interactions, that is van der Waals interactions and electrostatics. So these are non-bonded interactions. Now we will see one by one. So if we talk about bonded interactions, then it is the covalent bonding. If two atoms in the protein, if they are linked by a bond, then that means there is some bonded interaction which is taking place. Now what interaction can be possible? So first of all, the interaction which is possible is bond stretching. That means in a scenario when both the atoms, they are moving apart from each other, so in that case my bond gets stretched, or in other scenario they both can come close to each other. So in this way, that is known as bonded interaction, in which bond is involved. It is either getting stretched or relaxed. So all those things are happening. Second bonded interaction is the bond angle bending. So it involves three atoms. If there are three atoms, then it is possible that there might be some change in the angle between these three atoms and these two planes. So that is known as angle bending, because in this also these three atoms, they are linked by bonds. So it comes under bonded interaction, and we study that how the angle is changing with time. Third interaction is torsional rotation or dihedral angles. So for studying torsional rotation, we require 4 different atoms, because it is the angle which is formed between the two planes. So that's why we need 4 different atoms, and we study that how the torsion angles changes, because we have seen, according to Ramachandran plot, that there are certain phi-psi angles which determines that whether my protein will make alpha fold, or it will whether my protein will get folded into alpha helices or it will make beta sheet. So all those conformations can be determined with the help of these torsion angles, because they determine that whether what are angles which are feasible and which are not feasible. So these all comes under bonded interactions. Now the non-bonded interactions include van der Waals forces. So in this case, the van der Waals forces happens if the two molecules which are not directly bonded to each other, but there is some kind of interactions which is taking place. So in this scenario, if two of my atoms, if they come closer to each other, and if they come closer, means if the distance or the separation between them is very less, then in such scenario there will be some repulsive force which will take place, which will tend to push them apart. But if my molecules are far apart, then there will be some attractive forces which will act on them, which will tend to bring these two atoms together. So that is Lennard-Jones potential, which talks about both the repulsive and attractive forces. So in this case, because these are not directly bonded, these are the indirect interactions, so that's why they comes under non-bonded interactions, and these are the weak attractions. Then second is electrostatic or Coulombic forces. So these forces can act if we have the residues which are of different charges. So if amine group is positively charged and carboxyl group is negatively charged, So if those residues come closer to each other, so if those residues are negatively charged, then there will be some positive interactions, or there will be some attraction which will act. So that is known, that interactions, the charged interactions comes under Coulombic forces, where the charged molecule interact with one another. Then comes hydrogen bonding, which is also important. So these are also non-bonded interactions. Not direct covalent bond is formed, but these are also important. And different common force fields are charm, amber, bromose, and OPLS. So this is about different force fields. Yeah, and this GIF tells you that where different bonded and non-bonded interactions are actually present. So this tells you about the bond. If there is some stretching which will be happening, so that is known as E bond. That means energy of the bond stretching, and this is present between only two atoms. Then bending is happening between these three atoms, and then among these four atoms the concept of torsion comes into picture. Then van der Waals interaction will take place between two far apart atoms such as this one and this one, and then electrostatic will take place among the positively and negatively charged residues if they are present. Yeah, and these are some of the equations. So if you are interested, you can see what are the different formulas. So R specifies the distance, theta is angle, then for dihedrals this is the, and then impropers is also one of the term. So in impropers it is also between four different atoms, but in case of impropers we consider it if we want to study the chirality of a molecule. So this is similar to dihedral only because it involves four atoms. So in some scenarios we involve the impropers also into the potential energy calculations during the chiral cases. Yeah, so this also I found in another paper in which they have talked that how to decide which force field to choose. So choosing the right force field is very important in molecular dynamics simulation because it will determine that what results will you get. So first case or first thing which we should focus on is the accessibility. So there are some force fields which require a license. So in such scenarios we'll be, because the freely available force field is accessible, so we need to proceed with that force field. And some force field may be implemented in multiple software packages while others may be specific to one program. So for example, if you are using GROMACS to perform MD simulations, then there is some limitation. Only certain limited number of force field can only be used there. So that is also limiting your accessibility. So based on accessibility, you will choose the force field. Then performance. So we need to check if the force field reproduces the expected structure, stability and dynamics of the system. So if it agrees with the experimental data, then we can select that particular force field. Then comes community and validation. So before doing anything, firstly we'll look into the literature, that what other scientists are doing, which force field do they recommend. So based on that community and validations, which if somebody has already validated some force field, then we can decide from there as well. Then nature of the system. So it depends that what you are simulating. So based on that also you can select a particular force field. Then accuracy and efficiency. So generally we'll select those force fields which are highly accurate, which comes at the cost of computational power. So there might be some force field which require a lot of computational power. So in that case you cannot go for that particular force field because it will be a time consuming and costly as well. And then you will also see that it should be efficient as well as it should be accurate. So you need to take a balance between both of them. Then come simulation goals. So each force field is validated against specific experimental data. So make sure that it aligns with your research goals. Then computational resources. So force field range from simple and fast to complex and demanding. So you have to decide whether you want a simple force field or you want a fast force field. So it depends. So these are different scenarios which we consider while we choose for the force field. Now we'll talk about time and size scales in MD simulations. So this is the time scale in x-axis, and towards y-axis we have the length scale. So if we talk about bond vibration, so it's taking place between femtoseconds to picoseconds. So this is the time. If you want to study the bond vibrations, then you need to perform MD simulations which are at the time step of femtoseconds or picoseconds. Because if you consider the step size of milliseconds or for example seconds, then you won't be able to capture the bond vibrations. It appears that there is nothing which is taking place. So to study different bond vibrations, we need to perform MD simulation at this particular time scale. Similarly, if you want to study alpha helix folding, then this is the time scale at which the folding is taking place. So if we study MD simulation at this time scale, then only we'll be able to track that how the folding is taking place stepwise. Similar for the other kind of interactions such as side chain rotations, beta hairpin folding, loop motions, or protein folding. Because mainly we perform MD simulations to study the interactions as well as how the protein is getting folded. So that's why to study all these things, we need to decide that what time scale we want to consider. Yeah, so this is what we have seen here. And then size. So if you want to study atomic level interactions, then the size is at the level of angstrom. For protein ligand binding, the size is about at the level of nanometers. And for the large biomolecular complexes, the size is of micrometers. Yeah, so this is about the size scale. In this also I mentioned, so QM is the quantum mechanics simulations at the level of electrons. Molecular dynamics simulation acts at the level of atomic details, and this one is fast. Then comes coarse grained models, in which we study whole proteins and viruses, how they are changing conformations with time, and the biological experiments taking place from milliseconds to seconds, such as protein folding, translation, transcription. So all those things take place in this time period. Okay, now we'll see workflow of MD simulation. So we have looked into the overview of MD simulation. Now one by one we'll see the workflow. So this is the overall workflow, this slide. So in which firstly we need to obtain a valid structure, which could be experimental or theoretical. So from PDB you can get the protein three-dimensional structure. Then structure repair. So what happens in PDB? When somebody submit a structure, it is possible that there are certain residues for which we were not able to model the structure. We were not able to find the structure. So in that case, there might be some residues which are missing, for which we do not have the structural information. So we need to repair the structure. We need to fix those residues, and we also need to fix the steric clashes. Third step is structure preparation. So in which we remove the extra molecules and we add the solvent. So from PDB, in some scenarios, we get the complex structure in which the protein is already attached to some other ligand or some other substrate to which it is attached to. So in that case, we can remove those extra molecules and we can simply take the protein structure to model it. Then we can also add solvent, in which we add the water molecules to reconstruct the native conditions, such as the aqueous environment. Then assign the force field parameters, in which we decide that what are different force field parameters we want to study. So which we have discussed, bonded and non-bonded interactions. So all those things we determine that. Then energy minimization, in which we correct the bad contacts. Then comes equilibration. So before analyzing, we need to equilibrate the system, and ultimately we perform production run, in which we capture the molecular motion. So till here we are kind of preparing our system, we are kind of preparing our protein, we are stabilizing it so that we can perform analysis at different scenarios or in different conditions. So production run is the main thing, in which we capture the motions or we capture the dynamics. And after getting the dynamical picture of the protein in different time scales, we perform post simulation analysis, in which we extract the RMSD, RMSF and interaction data. So all these concepts we'll clear one by one. Yeah, so this is also the flowchart which tells you that what are the different steps. So firstly, structure preparation, in which we get the protein data bank structure. You can also get the structure from experimental results. Then we select the chain or the molecule. So for example, if I have a protein which is a dimer, trimer or tetramer, so in such scenario I can select one particular chain. Then structure check, we check for the structure. We visualize and check it by opening it, that whether all the residues have the coordinate positions or whether there are some missing residues. So all those things we validate. Then structure alteration. So for example, if I want to study different mutations, that how particular residue mutation brings about the change in the geometry of the protein or its conformation. So in that case, we can alter the structure. We can perform the point mutations using Wincoot, or we can also change the protonation state. Then preparation of simulation system, in which we solvate it, we add the solvent, we set salt concentration. So if my protein is charged, then we can make it stabilized, adding some salt, Na positive and Cl negative ions, so that the charge is balanced. We select simulation protocols. We prepare the simulation files. So in this case, we define the force field and all those protocols need to be completed. Then comes simulation run. Now after equilibrating and all those things, we perform the production run or simulation run, in which we see in live that how with time it is changing the conformation. So that is live analysis. We run the simulation and we copy the files to supercomputer or to which system you are working on, and in the end we perform analysis. So there are different measures on which we can analyze the conformation of the protein, its dynamics. We can study how it is changing. So now one by one we'll go through each step. So first step was preparing the molecular system for molecular dynamic simulation. So step 1 is to obtain a valid protein structure, which we can get from experimental data such as X-ray crystallography, NMR, cryo-EM, or we can get the theoretical data. So theoretical protein structure can be obtained from homology modeling or ab initio predictions, and along with this, AlphaFold can also give you the predicted three-dimensional structure. So that is not experimental. So these are different sources from where you can get a valid protein structure. Now the challenges with this structure can be missing residues or loops, incorrect protonation states, and unresolved steric clashes. So it is possible that your protein have a disordered residues, lots of glycines or lot of repeats which do not take a proper folded form. So in such scenarios, those residues will be missing residues. They do not have a proper three-dimensional structure or atomic coordinates. So those things, they are the challenges which we need to solve. Now second step is structure repairing, in which we identify and fix those missing residues or atoms. So correction involves correctly modeled residues or groups. So the residues which are incorrectly modeled, we repair them, we correct them. Protonation states of amino acids, so we also change those states so that we can stabilize the system. So protonation state is nothing but it tells you about the charge or pH, you can say. So rotamer conformations, so we can also change those conformations, and steric clashes. So we can see where the steric clashes are present, so we can try to solve those steric clashes. So for example, missing residue methionine 17 is repaired using the Wincoot software. So in this example, the 17th residue, it do not have the structural information. So in PDB file, we do not have this 17 residue with us. So what we can do, we can go to UniProt and we can figure out the protein sequence of this particular protein. So with this, we can identify what is the residue which is missing. So with the help of Wincoot, we can refine the structure. We can add this methionine 17 and we can see that how the structure is changing. So in this scenario, we make sure that our structure do not change. We simply insert the methionine 17 into this missing position. Or in the other cases, we can also check on the basis of electron density map that whether it is stable or there are some steric clashes. So if there is some steric clashes, then we also make some refinement using Wincoot such that the protein structure which we have obtained, it aligns well with the energy density map. So those things need to be kept in mind. Then generating input files. So we convert this PDB file into simulation ready format. So PDB file is what we put into our MD simulation software, but MD simulation software needs some simulation file. It directly do not work on the PDB file, so we need to make some changes. We generate topology file which describes bonds, angles and dihedrals, because we have seen that how we study dynamics, so we focus on different force fields, we focus on different interactions. So before moving on to them, we need to specify that currently what are my bonds, what are my angles and what are my dihedrals. So all these things are listed in the topology file. So from PDB file, you need to generate this topology file. In this, along with these informations, you also have atomic charges and connectivity. So what are the different interactions other than the bonded interactions what is present. So that will also be given in topology file, and we will also assign the force field parameters. So this comes under preparing the molecular system for MD simulation. Now the main thing is force fields and simulation parameters. So force fields are the mathematical representations of atomic interactions. So these are all possible interactions which can take place in my protein, because different atoms are bonded to each other. So what are different things which can happen with time? There can be stretching which can take place. So stretching, for example, if we have a spring, so if two atoms are attached with the help of a spring, then we can stretch it. The bonds are not fixed, they are not rigid, they are like spring, they can get stretched and relaxed. So in this way, this can also take place, bond stretching can take place. Angle potential, so it is also possible that my two atoms can move either closer to one another or farther from each other. So in such case, the bond angle between these three residues or these three atoms can change, such as hinge. So for example, if you have a laptop, you might have seen that you can flexibly open it, close it, so that is known as hinge. If you can move it, it is fixed at one side, but you can bring those two sides closer or farther from each other. So there is change in the angle between both the sides. So that is known as hinge. Then proper dihedral potential. So this is known as proper if you talk about dihedral angles. And if we talk about the improper dihedral angles, in which the orientation is not same as that in case of dihedrals. So these are two planes, and dihedral is the normal which is drawn to these two planes. So the angle between them is known as dihedral angle. So this also we study under force field. And then improper dihedral potential. So these all are the bonded interactions. And improper torsions, they are important for chirality. And also keep this thing in mind that to study these interactions we need at least 4 atoms. With 3 atoms we cannot study the torsion angles. Then comes non-bonded interactions, in which we talk about electrostatic potential and the van der Waals forces. So in van der Waals forces we talk about the Lennard-Jones potential. So electrostatic potential will work if we have two oppositely charged molecules. So in that scenario, if we have charged atoms, then only the electrostatic potential will come into picture. And as we know that in protein we have positively as well as negatively charged amino acids, so it is possible that we'll see the electrostatic potential taking place in a protein. So this is also we can study. Then comes van der Waals interactions. So this is the repulsive Pauli's exclusion principle, which talks that if two atoms are brought enough close to each other, then there will be some repulsion which will take place. And then attractive van der Waals talk about that if my atoms, if they are not very close and if they are sufficiently close to each other, then they tend to attract each other. So that is known as attractive van der Waals. So when we talk about van der Waals forces, we consider both these cases, attractive as well as repulsive Lennard-Jones potential. So these are different types of interactions which can take place in my protein or my biological molecule. So we study these things. These are the force fields. So with time we study how these interactions get changed. These are the measure with which we can compare. Third step is solvation and system neutralization. So in solvation we now firstly let's see why we perform solvation. So most biological molecules, they function in the aqueous environment, because in our body it's 70% of water, so proteins are present in the aqueous environment. So that's why we need to perform solvation, in which we add water molecules or solvent with our protein. Then types of solvent models. So there are different models because we are performing it in computational methods, we are performing it in a computer. So we need to decide the model. So first is explicit solvent model, which involves, so the examples are tip 3P, SPC, tip 4P. So whenever you use some MD simulation software, so in that case these are different solvation models. So you select any one of them. They comes under explicit solvent model, in which individual water molecule is included and it is computationally expensive. So this one is explicit solvation. As you can see here, the protein is present here and water is uniformly present throughout the system, in which each water molecule will be considered. So this is explicit model because you are considering each water molecule, so it is expensive, it will take time to perform different calculations. Then comes implicit solvent model, which was given by generalized Born and also is based on Poisson-Waldman. So it is a solvent treated as a continuous dielectric medium, faster but less accurate. So in this case, the entire water molecules, they act as a medium. Instead of considering each and every water molecule, you consider it as a medium. You do not perform calculation of individual water molecule. So in that way, this is quite quick, but it is less accurate. So we need to make a balance. Depending on our conditions, we select any one of the solvent models. Yeah, so in implicit, water is only included implicitly as an effective or averaged interaction with no atomic details. Whereas if we talk about explicit solvation, then it is the representation of water is included at the molecular level. And then what this solvent does, so viscous dampening force, dielectric screening or polarization, hydrophobic effect, and also structure. So it maintains the structure, it provides the native like situation, so that my protein can get into my native conformation. We try to make the conditions as possible, means as much similar to that of the native state conformation. So that's why we perform solvation. Next step is to add the ions for charge neutralization. So now why to perform this neutralization? So we know that our proteins are charged. So simulation box which we make, that must be neutral. So how to neutralize the protein? We add the counter ions. For example, if my protein is negatively charged, then I will add those number of Na positive ions that can stabilize the negative charge. We'll see what is the charge on the protein, either it is 2 minus or 3 negative or 4 negative. So based on that, we will add those number of Na positive ions, or vice versa, Cl negative if my protein is positively charged, to balance the system charge. Now we adjust the salt concentration for physiological relevance, so that also it does. So in this scenario, we are performing neutralization. Third step is energy minimization. So now in native state, in our body, in vivo conditions, the protein is present in its most stable form. So if you want to perform MD simulation, then you need to make sure the protein with which you are starting, it should be in its stable state. Then only the comparisons are helpful. If you are already taking the protein which is not stable, which is unstable, so in that way you will not be able to understand the dynamics. So for those cases, we need to minimize the energy. So it removes the steric clashes, it achieves the energetically favorable conformation, which is supposed to be most closest to my native conformation. The common algorithms which we generally use in this case, they are the steepest descent and conjugate gradient. In steepest descent, it is fast but it may not converge, and in conjugate gradient, it is slow but more precise. So we'll see these algorithms also, so don't worry about it. Firstly, we'll talk about free energy of solvation, because in this scenario we are adding water molecules, so there is some energetic changes which are taking place. Because now, before moving on to energy minimization, we'll also see that if we perform solvation, then does it bring about change in the energy or not. So in this case, delta G sol, that means the energy of solvation, it is the free energy change when transferring a molecule from vacuum to solvent. Because in your system you have loaded a PDB file which was in vacuum, now you are moving it to a solvent, you are adding water molecules, so there will be some free energy change which will take place. So this delta G solvation is a sum of delta G that means electrostatic component, delta G van der Waal, that is van der Waal interaction between solute and the solvent. So solute is your protein and solvent is the water molecule which you are adding. So different interactions will take place, different energy terms will come into picture. So electrostatic interactions will take place, van der Waal interactions will take place. Then delta G cav, which is free energy required to form a solute cavity, which includes the entropic penalty due to solvent reorganization. So this, after adding the solvent, there might be some reorganization which will take place, because protein will tend to make interaction with the water molecule. So delta G K will also come into picture, and then this is delta GHP, which is the explicit hydrogen bonding term. So there will be, after forming the hydrogen bond, this will also bring about change in the energy of the system. So that's why understanding the free energy of solvation is important, because it can bring about change in the total energy of my system, because I'm moving it from my vacuum state to a solvent or solvent state, aqueous state. Now we'll talk about energy minimization. So firstly, what is energy minimization? So it is a computational technique which we use in MD simulations to find a stable and low energy conformation. So why stable and low energy conformation? Because the protein in its native state, it is in lowest energy form and it is highly stable. We reduce steric clashes. That means if atoms they are too close to each other, then we tend to we refine the structure so that it should have least steric clashes. Then we ensure realistic starting structure, because in the MD simulation you are recreating the actual conditions in the computer. So you need to make sure that the starting point, it should be as much as similar to my native state. So key objective is to find a local energy minimum in the potential energy landscape. So I hope you are clear about the potential energy landscape. So it tells you about the energy of your system or energy of your protein. So in the potential energy landscape, you will see that where my protein isn't currently present. So either it is present in a high energy state, so we tends to minimize the energy, and we'll see that what should be my extreme energy point, which is the lowest energy point. So from there I can stop energy minimization and I can proceed with the simulations. So now why is this energy minimization necessary? So real world experimental structures, they often have bad context due to missing residues or incorrect modeling. So for that reason we need to perform minimization. Biological molecules must adopt physically stable conformation before simulations. So for allowing my protein to get a stable conformation, we need to make sure that it should be in a least energy state, which is also stable. Helps avoid high energy unstable configurations that can cause simulation crashes. So for these reasons we go for energy minimization. So for example, if two atoms are too close, then unrealistic repulsive force will appear, which will lead to simulation instability. So that's why we have to go for energy minimization. Potential energy landscape. So energy minimization process, it moves the system to lower energy state. So for example, if my protein is here, let's talk about this figure. So this is the potential energy towards y-axis, and this is the variation in parameters. So we vary the parameters and we are starting from this point. So we are moving down in the potential energy axis, we are moving down to a lower energy state. So in this scenario, this is a lower energy minimum or local energy minimum. Then when we were talking about the Monte Carlo simulations, so in that case we have studied that it is possible that my protein, it might get trapped in a local energy minima, which is not the global minima. So global minima is the one which is the most stable form, means below which we do not find the protein below that energy level. So this is my most stable form, this is the global energy minima. So in certain scenarios my protein can get trapped in a local energy minima. So in those scenarios we need to provide some external, we need to rise the temperature and some external energy is provided so that it moves to another point by overcoming the energy barrier, and from this state it can either move towards the right side or left side. So if it moves towards the right side, then it will further move downhill to a lower energy, lowest energy state, which will be my global minima. So our ultimate aim in energy minimization is to move our protein from the starting point or the high energy state to the lowest energy state, and we try that it should reach the global energy minima. Although in certain scenarios, based on the time it takes and your knowledge, you might get trapped in local energy minima. So it depends. So means our ultimate aim is to go to the global energy minima. Yeah, so local energy minima, it is a stable, but it is not necessarily the lowest energy state, whereas global minima is the absolutely most stable conformation. So our ultimate aim is to go to global minimum, but it is difficult to reach. It might take a lot of time. So now we'll see that what are the different algorithms with which we can reach to that level. And one more term which I want to introduce is the molten globule states. So molten globule states are the states where the protein is trapped in such means in that particular energy level, which is not the least energy level. So somewhere middle, not very high, not very low, but in some middle state when my molecule is trapped, that is known as molten globule state, which means that we further need to minimize the energy. So that state is molten globule state. Then comes transition state, after which the transition happens because my protein get properly folded. Till molten globule state, my protein is not properly folded, it might be unfolded in some points. So this is the glass transition, after which my protein will be properly folded and discrete folding intermediates exist, and ultimately we reach to the native state, which is having the least potential energy. Yeah, so now there are different energy minimization algorithms which we use to move our protein from the initial point to the point which is the global minima. So first algorithm is steepest descent algorithm. So it moves the system along the steepest gradient. It is fast but inefficient for deep minima. So if we have a deep minima, then this method will take a lot of time to reach here. So that's why it is inefficient if we have a deep minima. If we have narrow minima which comes after 2-3 steps, then this method can be used. But although it is fast, it is suitable for initial relaxation of the system, and it converges quickly for small steric clashes. So if you have small steric clashes, then it will converge quickly and you will be quickly reaching to the global minima point. So this is the equation which talks about so these x specifies the atomic position, because your protein is present in a three-dimensional space, so it has some position, it has xyz coordinates. So this talks about x k plus 1, that means at step k plus 1. So this is the x is the position at step plus 1 is equals to xk. So that is the position at step k. So when you are moving from one position to some other position, then you are taking some step. So it's like a step. So from here to here, it is taking one step. So this is my k and this is my k plus 1 when I am taking one step down the energy gradient. So in this scenario, the atomic position at xk plus 1 is equals to xk minus alpha k. Alpha k is my step size or the learning rate. So it depends how much longer step I am taking, whether the step size is large or it is small. So this is generally constant, which is fixed. And then partial derivative of energy at xk, because in this scenario I am moving to a lower energy state, so I will make sure that the energy at point x, when I am performing the partial derivative, then I am making sure that the energy which is at step xk plus 1, it should be low. So overall this xk and xk plus, so the energy at xk plus 1, it should be lower than the energy at xk. So that's why this energy term is considered. This is the gradient of potential energy function at step k. So in this way we are considering the gradient of energy while when we are moving from one step to next step. Then for next step, whether we need to take that step or not, we will perform this energy gradient calculation. We'll see that what was the energy at step k. So this will help us to determine that in which direction we need to take the step forward, whether we need to proceed with xk plus 1 this step or some other direction we need to take a step. So this is known as steepest descent. And one more thing is that it adjusts the atomic positions in the direction of negative gradient to minimize energy, but it can be slow. So it's known as steepest descent because it will move to the direction towards the negative gradient. So it will move towards that direction where my energy gradient is decreasing and not uphill. It will always move downhill. Then comes next algorithm, which is the conjugate gradient algorithm, which is more efficient than steepest descent because it uses the information from previous step to guide the descent, and it works well for the complex systems with deep energy wells. So in the previous case we have seen that if there are deep energy wells, then it is taking single multiple such steps, and it is taking lot of time. But in conjugate gradient, what it is doing is it is taking larger steps, so it is quickly converging. So that's why it is good for these deep energy well as well. And this is the equation. So at position at k plus 1 step is equals to xk plus alpha k pk. So initially in place of pk we had partial derivative of E at xk. So now we'll see what this pk means. So pk, it is the search direction, because it is telling you the direction. So in this case, this direction will be negative partial derivative of energy at xk plus beta k pk minus 1. So as we have seen that it is also taking consideration of the previous step. So in this scenario we were only considering my k step, but in this scenario we are also focusing on xk minus 1. So that means I'm also taking into picture what was my previous energy. So that's why this is much more better. So beta k pk minus 1, and the pk is, this is the formula for the pk, and now beta k is the partial derivative of energy. So in this case, instead of only considering this previous step, we are also considering the step previous to that step. So for example, moving from this second point to third point, instead of only considering this energy gradient of second point, I am also considering the gradient of my first point. So that is what conjugate gradient algorithm tells you, that not just one, but it also takes the previous step also into consideration. That's why the convergence is quite high, it's quite quick, because it is taking larger step size. So it efficiently minimizes the energy by combining current gradient with previous directions. That's why it avoids the redundant steps. So in this way it will not move to other direction, it will based on the previous step, it will take the wise decision without redundantly moving left, right. Then the third method is Newton-Raphson method, which is the fastest among all, and quadratic convergence for well-behaved functions. It uses both the function value and its derivative to refine the approximation, and it works well for the functions that are smooth and differentiable. And it is also efficient compared to other methods like bisection and secant, and it requires the initial guess which is close to the root for successful convergence. So I know these terms might sound a little bit more mathematical, but you do not have to worry about it. So in this scenario, at step n plus 1, it will take into picture xn. Now what extra it is including is the hxn inverse, which is my Hessian matrix. So it is the inverse of Hessian matrix. So my Hessian matrix is the second derivative of energy function. So till now, the two methods which we have seen, steepest descent and conjugate gradient, so in those two methods we were only considering a single derivative, but the difference in this method is it is also taking into picture the second derivative. So second derivative is calculated using the Hessian matrix, and the second derivative is of the energy function. So what the second derivative means? So first derivative means with respect to one variable you are studying the change in energy. Second derivative means if you are considering other than one scenario, or other than means with respect to only one condition, you are also looking for the changes with respect to some other condition as well. That is known as second derivative. That how with respect to my other point, how the energy is changing. So it represents the curvature of energy landscape with respect to atomic positions. And the inverse is that inverse of Hessian matrix, which is used to account for curvature of the energy surface. So that's why one add-on thing with to other method is the second derivative. So Hessian matrix is taken into picture, and along with this partial derivative of energy is also taken into picture, where we are fixing the position. So xn, that means at previous step what was the energy term. So if this derivative for xn plus 1, it should be negative, to make sure that we are moving towards the negative energy scale. We do not want that our system moves from high energy state to higher energy state. So our aim is to move from high energy state to lower energy state. So partial derivative of energy should be negative, and along with the partial energy state, we are also considering second derivative. Now if we break down this, so energy gradient, which is the partial derivative of energy at position xn. So this gradient represents the direction of the steepest increase in energy, and the negative gradient points to the steepest decrease, or the direction of descent. So direction of descent means to which direction you want to move. Then energy Hessian, which is hxn. that is Hessian matrix. So in this case we are using inverse of this. So the Hessian matrix contains the second derivative of energy with respect to atomic positions representing the curvature of energy surface. So it tells you about the curvature. And then update step. So Newton-Raphson updates uses both the gradient and the curvature. So gradient means force, how my energy is changing, and along with this how my curvature is changing, to make sure that I am moving towards my negative curvature. So I am moving in depth instead of moving in the flat surface. So that is about the curvature. To adjust the atomic positions, the inverse Hessian scale the gradient to optimize step sizes based on the energy surface curvature. So step sizes are also very important, because if you will take small steps, then it will take a lot of time, and if you take very large steps, then also you might miss on the your global minima. So in that way you need to make sure that your step size should be in between both of them. It should not be very large and it should not be very low. So in that way, in Newton-Raphson, because you are taking second derivative, so you are along with the direction, you are also taking into picture the size, the step size, because you are looking into the curvature as well. Yeah, to summarize, we start with initial guess for atomic positions. We calculate energy and its gradient. We compute the Hessian, which is second derivative of energy or curvature, and then we apply the Newton-Raphson update to iteratively minimize the energy. So among all these steps, Newton-Raphson is the quickest among all. Okay, so now you have minimized the structure. Now we need to perform equilibration. So equilibration, and ultimately we'll proceed with the production run. So equilibration, so why we need to perform equilibration? Because we stabilize the temperature, pressure and density before actual MD. So when protein is present inside our body, then it is in certain conditions. It is in some ideal temperature conditions, some pressure, and its density is also, it has some density. In the medium it has some density. So in that way we need to make sure that the conditions are similar of in vivo state. So that's why we need to stabilize. We need to find out the temperature at which my protein is highly stable. We need to find out the pressure at which it is having the lowest energy. It is in the most stable conformation. So in equilibration we tend to find out that temperature and pressure in which my protein is most stable, so that I can maintain those temperature and pressure conditions, and now I can study the dynamics. So there are two stages in which I can equilibrate my system. First one is NPT ensemble, which means when I fix my volume and temperature. So this is the condition where my temperature will be fixed, my volume will be fixed. Now protein, it is allowed to undergo change in the conformation. It can change its density. So temperature is stabilized and it ensures that the system is properly thermalized. In NPT ensemble, the constant pressure and constant temperature will study. We adjust the system's density and we ensure the realistic pressure conditions. So this is about NPT and NVT and NPT. In the next slide, we will see in detail about NPT and NVT. But for now, this is the overview. Then comes production run, in which you will be running it. So the actual MD simulation will begin after the equilibration. The system is fully equilibrated and runs at constant conditions. Data collection will be performed after the production run, which will involve the collection of atomic coordinates, because by taking the atomic coordinates, now what you will do, you will compare it with your bile type structure. You will see that with time how my coordinate is changing. That will tell you that how the position of those residues is changing, either if in folding, because whenever my protein will get folded, then it will change the conformation. So with change in conformation, its residue position will also change. So with the help of atomic coordinate, that can be tracked, velocities and energies. So we can also see what is the energy of my system and with what velocity it is bringing about the change. There are certain proteins which get quickly folded, whereas other proteins take a lot of time. Similarly, we talk about the protein ligand interactions, such as drug binding to the protein target. Then that binding, in some scenarios that binding can take a lot of time, whereas in other cases the binding can be quick. And then after that binding, how much time it will take to bring about the conformational change. So in all these things can be studied, and interaction patterns can also be studied. So these are the graphs in which we have identified what is my constant temperature which I need to maintain to study my protein, and what should be my pressure. So we see that these red lines, it represents the fluctuations, structural fluctuations. So we make sure that the pressure or temperature which we pick up it should be that point in which these fluctuations are very less or minimum. So in that way, that particular pressure and temperature is selected, and later on the analysis are performed by making that particular temperature and pressure conditions constant. So firstly, we'll look into the NVT ensemble, that is constant volume and constant temperature. So it is also known as canonical ensemble. So keep this word in mind, that NVT means canonical ensemble. It keeps the number of particles, volume and temperature constant. So these three things are kept constant in this case. Now what is the purpose of this NVT? So it allows the system to adjust the velocities based on defined temperature. It removes the artificial high energy conformations, and it prepares the system for pressure stabilization in NPT. So firstly, this is the stepwise procedure. Firstly, we'll equilibrate using NVT, in which we make the volume, temperature and number of particles constant, and later on we will perform NPT, in which we will perform pressure stabilization using NPT. So till now we have not made pressure constant. So this is step by step. Firstly we'll make volume, temperature constant, and later on we'll make pressure constant. So with this, my protein which will be obtained after NPT and NVT will be highly equilibrated. It will be highly stable. So common thermostats which we use to maintain the constant temperature is the Berenson thermostat, which is fast temperature coupling, but it is less accurate, or Nose-Hoover thermostat, which maintains correct temperature fluctuations. So the equation in NVT is means the change in energy with time is 0, where V is constant, which is volume, and T is controlled, that is temperature. So significance of NVT is that it helps us to maintain the constant temperature. So we fix the temperature and simulate the controlled thermal conditions. Fixed volume, because in this we are also fixing the volume. So volume means my protein which is present in certain conformation. So when it gets folded or when some interactions take place, it can undergo change in its volume. Volume means how spread it is, how much surface area it is occupying. So that is known as volume. But in this scenario, we are maintaining constant volume. That means I'm not allowing my protein to move, my helices to move apart or performing different interactions or other things. So in this case, volume is also fixed. So in this way, this is useful for studying the systems in isolated or constrained environments. So isolated means there is nothing other which is present. So if there is some other residue, if there is some other substrate or ligand which is present, so it can allow my protein to undergo change in the volume. But in this case, volume is fixed. That means there is nothing which is present. This is a closed environment or isolated environment. So volume will not change. Energy minimization, so it helps to focus on internal dynamics. Energy minimization without pressure variations. So internally what is undergoing, that study we does with NVT. And applications are solvent protein interactions. So with solvent, how my protein is interacting, that interactions can be studied. Protein folding can be studied. Simulates folding in isolated environment where temperature is controlled. When temperature is controlled, then how the folding is taking place. So it is also possible that at particular temperature only, with time, protein gets folded. In our body, temperature is not changing very much. So that means at constant temperature also my proteins can get folded. So in this way we can study that condition, that how at constant temperature my protein is showing the dynamics. Ligand bindings. So model the ligand binding in a fixed volume setup with constant temperature. So volume is fixed. Then crystal structures, so suitable for simulating the proteins in solid phases where the volume remains constant. So in that scenario, we tend to make it like a little rigid structure. We are not allowing it to change the volume in case of NVT. Then comes NPT ensemble, in which we are now making our pressure constant along with my constant temperature. So this one is known as isothermal isobaric ensemble. Previous one was known as canonical ensemble. This one is isothermal and isobaric ensemble, which keeps number of particles, pressure and temperature constant. So the purpose is to adjust the density of the system to match real world conditions. So in this case, density is changing, we are adjusting the density. So that's why in this case volume can also change. It removes any vacuum region from the simulation box and it prepares the system for my production MD. So common barostat, because in this case I am making my pressure constant, so I will use a barostat. So Brentson barostat is used and Perinello-Rahman barostat can also be used. So this Brentson is fast pressure control, but it can distort the results, whereas this Perinello-Rahman barostat, it is more accurate and it is suitable for long simulations. So this is the equation where the change in pressure with time is constant, it is 0. Temperature is constant and pressure is controlled. So I will choose that pressure where the change with time is 0. And this is the curve. So density is changing with time, whereas my pressure temperature is constant. So this shows you that when we have performed NPTEL, then it is kind of getting close to each other. So it is increasing, so decreasing volume and increasing density. Yeah, so earlier the units were far apart, but in this case this vacuum is filled. Now the vacuum is filled with the solvent molecule. So this is what takes place in the NPT. So significance is that to recreate the realistic conditions at constant pressure and temperature, which is in the real biological environment as well. Volume fluctuations, so it allows the volume changes and it simulates that how my protein and solvent will adapt. So when I add a solvent, then protein can interact with the solvent and there are certain things which are taking place. So it allows for those changes. It allows for volume fluctuations and equilibrium, so it reaches a thermodynamic equilibrium which ensures the natural protein behavior, because with time it will, in the presence of solvent, there are some changes which are taking place. So that's why it makes sure that that is the natural protein behavior. So applications are to study the solvent protein interactions. Protein folding can also be studied, ligand binding and membrane proteins can be studied with this. So it models the membrane proteins in lipid bilayers, allowing the temperature and pressure fluctuations. So means for a particular single run you have fixed pressure temperature. For another run you can change those pressure temperature. We will fix it for that particular MD run, but those pressure and temperature values can be different from my first MD run. So those comparisons can be done in this case of MD simulations. And this is a difference between the NPT and NVT. So means, because I felt that this is important. So in NPT we have constant temperature and pressure, whereas in NVT we have constant temperature and volume. NVT is the canonical ensemble. That means we perform first NVT and then we'll decide that whether we need to perform NPT or not. So NPT comes after NVT. Now volume. So in NPT volume can fluctuate can change with pressure, whereas volume is fixed in NVT. So as in the name NVT. So VT means volume temperature fixed, PT means pressure temperature fixed. Then pressure, so pressure is constant in NPT, whereas in NVT pressure is not controlled. So that means it can fluctuate. Temperature control, so because in NPT and NVT, in both of them we maintain constant temperature. Volume control, so in the NPT we do not control the volume, system adjusts to pressure. So if pressure changes, volume can change, whereas in the case of NVT volume is fixed, so there is no change in the size of my system. System type, so it simulates the system where pressure and temperature are constant, whereas NVT simulates the system where temperature is fixed in a fixed volume. So applications are real world systems, liquid gases and membrane proteins, whereas in the case of NVT we look for the isolated systems. So that might not always be the case in the natural scenario, because I am making the environment isolated. I am not allowing my protein to undergo the change in the presence of the solvent or in the presence of the ligand, because volume is getting fixed. So in this way this is helpful in the case of isolated studies or fixed volume conditions. So if we talk about flexibility, so NPT in this volume and pressure can adjust based on the system needs, whereas in the case of NVT volume is rigid, so it cannot get adjusted to pressure or size. Then there was some paper in which they have mentioned that although the NVT is the canonical, but without this, means by considering only NPT, which is the high initial pressure, this is not always the case. We need to perform both of them. If we consider both NPT and NVT equilibration, then that gives us better results. So in this out of sight, out of mind, so they have studied the effect of the equilibration protocol on structure ensembles of charged glycolipid bilayers. So as these are MD simulations, so how you take different steps, what are the different things you consider that can make a change. So in this case, they have these are the key points through their paper. So they have seen that there are some challenges in simulating the LPS membranes. So LPS means lipopolysaccharide membranes. So they are studying membrane dynamics. So standard equilibration protocols for phospholipid bilayer, it can cause issues. So when applied to complex LPS membranes, including the unintended water intrusions. So these are some of the water intrusions which are taking place. So if we only perform NPT, then these water intrusions are present, which can give different results with MD simulations. So we should remove these water intrusions. So solution is to perform stepwise thermalization by using both NVT and NPT protocol. So the proposed stepwise thermalization prevents the water buildup and pressure fluctuations, ensuring more stable equilibration of LPS membranes. So in this way, they are telling that using both NVT and NPT stepwise can help you to get rid of these unintended water intrusions. So protocol validation and practical applications. So the new protocol was successfully demonstrated using this LPS, DPPE and lipid A membranes with different salts in GROMAX. So they have altered different salt conditions and they have provided a reliable and easily implementable approach for consistent simulation of charged glycolipids. So the molecule which you are studying, that is very important. So you need to first understand what molecule you are considering, so that you can decide what are the things you need to consider, what force field you should choose, how equilibration should be performed, what should be performed first, whether it should be a combined way in which we perform it stepwise, NVT combined at different steps, or individually we need to perform it. So all those things need to be kept in mind, because these little variations at the steps can bring about change in the final results of MD simulations. So now after performing the MD simulation, after performing the production run, now we are getting the results. So we have to perform its analysis. So this comes under post simulation analysis. So after the simulations completed, we need to analyze it, that how it changes. So first metric which we consider for the analysis is the RMSD, which is root mean square deviation, which measures the average deviation of atomic positions from a reference structure over time. So in RMSD we see that if we have a certain structure, then after performing simulation, how the structure gets changed. So from initial structure to the final structure we make the comparisons. So this is the formula for RMSD calculation, which is the square root of 1 divided by n. N is the number of atoms. Then this integration, so from 1 to n, this is RIT, which means position of atom I. So these are the atoms ranging from 1 to n. For each atom, we will perform the square of the difference of the position of atom I at time point t, and then its reference position. So ultimately in RMSD we check that how my position of atom I, that means position of all these atoms, changes with time. At time t, what was the position and what was the reference position. So this difference, we perform square of this difference and take the square root for all the differences ranging from I number of atoms to, means from I1 to n number of atoms. So this is known as RMSD. If your RMSD is high, that means there is higher distance, there is more separation, that means the structure which is predicted, that is variable, or the ultimate conformation which you have obtained after MD simulation, so it varies a lot from your initial structure. So if RMSD is high, then it tells you that. Then comes RMSF, which is root mean square fluctuation. So this measures fluctuation of each residue or atom over the trajectory, in which the formula of RMSF for I atom is square root of, so previously RMSD, it is at time t, so this is for the entire protein. In this we consider residues, means atoms from 1 to n, whereas RMSF is given for each atom. So for RMSF for I atom is the square root of 1 upon t, which is the total number of time frames, and summation t1 to t, that means from first point, the first frame to total number of frames, that is capital T. We will find the square of the difference between Ri, that is position at time t, to the position at, that is the average position of atom over time, that is Ri. So we see how much it fluctuates at different time points, so that is known as RMSF for that particular I. So if there is some fluctuations that is taking place, that we can study, because with time it is possible that there are some interactions taking place, so my particular atom can change its position. So that is known as fluctuation, that how much it is varying from its initial position with respect to time. Yeah, so this is in detail about RMSD. So it quantifies the structural stability of a molecule by measuring the average displacement of atoms over time. Lower RMSD means you have a stable structure. That means what you have initial structure, your final structure after going through a certain time period, it is still the same. So that means it is stable. There is not much change in the atomic coordinates. Whereas if the RMSD is high, then it means there are some significant conformational changes that have taken place. And it is used to compare the simulated structure with an experimental reference. And if we talk about the root mean square fluctuation, then it measures the flexibility of individual residues. If you have high RMSF, then it indicates that the structure is highly flexible. That means certain residues, they are highly flexible. They are changing their position with time, or mobile regions, or those are mobile regions. So what happen is that if you have an enzyme, then as soon as it binds to substrate, there is some kind of, according to induced fit hypothesis, after binding to a particular substrate or ligand, the protein can undergo change in the conformation. So to study those changes, those fluctuations, we can use RMSF. So if the RMSF is high, then that will tell me those atoms or those residues which are highly flexible. So it can help me to study, to understand which of the region in the full protein are highly flexible or are mobile. Then it is essential for identifying the active sites or functionally important flexible regions in the proteins. So it is also possible that in active site the structure can get changed as soon as my protein binds to ligand, but along with this there is a possibility that my substrate is binding to active site and it is also bringing about change in the conformation of the protein at some other site. So that thing can also take place. So in this way we can understand those fluctuations. So this figure is adopted from another paper in which they have studied RMSD and RMSF. So what this figure tells you? So this tells you that how my RMSD is changing with time. So x-axis is my time point and RMSD is my distance between the positions of my atom, means my atom from my protein at current state versus the reference. So how much it changes. So as you can see in this, at time 140 nanoseconds, the RMSD value is high. So this tells you that there is something different which is taking place. There is some change in the conformation which has taken place at 140 nanoseconds. So that means there is some transition or unfolding event taking place due to which the RMSD is increasing. Whereas if you look here, which is the RMSF plot, so this shows you residue numbers with respect to RMSF, that how much fluctuation is taking place and at what residue position that fluctuation is taking place. So in this you can see at nearly at 150 to 175 there are some peaks which are observed, whereas somewhere there is not much peak. So at residue position 100 the RMSF was close to 0.1, whereas here the RMSF value is high between 120, means at 135 to 175. So this means these are the residues which are mobile, which are showing higher fluctuations. So these analysis can be done by considering RMSD and RMSF. They can tell us some novel insights, that which residues are highly fluctuating, so that we can focus on those residues and see that why they are changing the atomic positions, what is important. So After performing the RMSD and RMSF calculations, they have performed the overlay of structural conformations. So from 0 to 200 nanoseconds, they have taken picture of protein conformation. So what they do is now that for each such position, they are overlaying it. They are overlapping all those pictures of the protein which they have taken, and then they are seeing that which positions are showing the change in the conformation. So here they have pointed out that this region is highly flexible. So when we are overlaying, then this region is showing multiple such lines instead of overlapping on each other and showing the darker side. So there are multiple thin lines which are observed. That means these are not properly overlapping on each other. So that means this region is mobile. It is changing the conformation. So this, after identifying the residues, they observed that threonine 159 to lysine 167, there is a loop. So this loop is highly flexible. That's why this is not properly overlaying on each other. So in this is to show you that how the RMSD and RMSF calculations can help you to analyze the protein. This will give you the novel insights. Now the third parameter is radius of gyration, which represents the overall compactness and folding state of a molecule. So if it tells you that either your protein is compact or it is not compact and it is spread out. So because if protein is compact, that means it is fully folded, it is properly folded. But if it is not compact, that means it is unfolded. So decreasing radius of gyration suggests that protein is folding. So with time, for example in this case, they are measuring that how the radius of gyration is changing with time. So for all these three cases, we can see radius of gyration is kind of decreasing with time. That means my protein is getting folded. And if we compare among these three cases, P173A, B and C, then you will see that for my P173C condition, which is my green, it has lowest radius of gyration as compared to 173A and B. So if you start from here, then you will see it has the lowest radius of gyration, which means this particular protein is most stable and it is highly compact. Then comes 173A and ultimately 173B is least compact among these two, and it fluctuates significantly. So its radius of gyration is constantly changing. So which means it is getting compact, then it is opening up, then it is again getting compact. So that means there is some fluctuations which is present, whereas in both the green and blue, there is not much fluctuation as much as you can see in the case of red. So this means this is highly flexible protein, whereas these two, they are compact and folded, with blue one showing minor variations. And this is the formula to calculate radius of gyration. So this takes into consideration total number of atoms. So for all the atoms, we calculate the radius of gyration, where m is the mass of atom, r is the position of atom, and rcm is the center of mass of the structure. So center of mass is identified for the entire protein. You identify what is the center point. So if you have the center point, then you calculate the distance from that center point of from all such residues which are present. We calculate how much far they are from the center point. So if they are close to that center point, that means the structure is compact. If they are far from the center point, then structure is open up, it is unfolded. So that is what radius of gyration tells you. Now the next metric which we consider is the hydrogen bond analysis, which identifies and quantifies the number of hydrogen bonds which are present between the molecules or within the molecules. So if the hydrogen bonding is strong, then that means the structure is stable, because we have seen that secondary structure formation, whenever the protein takes it converts to secondary structure form, then there are hydrogen bonding which support that secondary structures, alpha helices, beta sheets. So that's why if the hydrogen bonding is strong, then my structure will be stable, it will be having the structural integrity. It is also essential in drug binding studies because hydrogen bonding play important roles in molecular interactions. So these are non-bonded interactions, but they are very important for the stability. So this is the formula to calculate hydrogen bond. So if there is no hydrogen bond, then it is 0. Otherwise, there is some distance. So if distance between two atoms is less than 3.5 angstrom, then there is a possibility that there can be hydrogen bond that is present. So this distance cutoff is important. So if the cutoff is less than 3.5 angstrom, then that means there is hydrogen bonding interaction present. Otherwise, there is no hydrogen bonding interaction. And the most important thing which I have not mentioned yet, that hydrogen bonding will only be present if there is hydrogen that is present. So whenever hydrogen is attached to most electronegative atom, then it interacts with the another hydrogen that is electronegative. So in such cases there is the hydrogen bonding interactions which take place. Yeah, so you will be finding the distance between those two residues. If there is no hydrogen, then hydrogen bonding is not possible. You cannot find the hydrogen bonding interaction between two oxygens or two carbons. So hydrogen must be present. So there must be one hydrogen bond donor and must be another should be hydrogen bond acceptor. So we calculate the angle. So that cutoff angle is 120 degrees and distance between those two should be 3.5 angstrom. So if that conditions are satisfied, so that means you have the hydrogen bonding interactions present. If more hydrogen bonds are present, that means structure is highly stable. So in this case there are three situations. One is HSP90 AA1, which is aleone A, and then AKT1 and AKT3, which is represented in green. AKT1 is black and the red one is the HSP90AA1. So this shows the number of hydrogen bonds. So there are number of hydrogen bonds towards y-axis and x-axis is the time. So these are the picosecond time period. So we can see among all these three, red one has the most hydrogen bonds, which are about 6 to 8. So which means it is stable throughout. The number of hydrogen bonds is maintained constantly throughout time. Whereas if we talk about this black, then there are some hydrogen bonds which are observed near this time period, then there are no hydrogen bonds, and then again hydrogen bonds appear, and then we again loses the hydrogen bonds. So which means that this is less stable because there are fluctuations in the hydrogen bonds. So that means it is undergoing change in the interactions with time. Then if we talk about this green one, then there were no initial hydrogen bonds and ultimately in the end hydrogen bonds appear. That means after this time point it is showing the interactions, it is making up hydrogen bonds. So all these interpretations we can make after looking at the number of hydrogen bonds with time, that how the hydrogen bond interactions are coming into picture. So how we are able to capture this? Because we have the atomic coordinate information for each atom. So by looking at those atomic coordinates, we find out that my two hydrogens present in my protein, what is the distance between those two atoms, and then we figure out that whether it can make a hydrogen bond or not, and after that we calculate the number of hydrogen bonds at each time point and figure out that how many number of hydrogen bonds are present. Yeah, so if we talk about this first one, red one, then it has strongest interactions. The black one has the weaker interactions or weaker binding, and green one has the delayed interactions which appear at a time point of 40,000 picoseconds. Now next analysis which can be performed is the PCA, which is principal component analysis. So it tells us it gives us the idea about the protein backbone motion along the MD simulation trajectory to understand that how the folding or loop movement is taking place. So instead of considering one single residue or instead of considering entire protein in PCA, what we can do is we can consider a backbone, protein backbone, or we can consider a loop, certain substructure present inside a bigger protein. So in that way we can compare the motions. So motion is mostly described by first two eigenvectors and eigenvalues. So these are the mathematical calculations which are performed. So after obtaining the atomic coordinate information at different time points, we will compare the eigenvectors and eigenvalues and we will see that how much they are varying. So if we talk about eigenvectors, so they represents the direction of maximal variance in the protein's motion. So because at time period you are looking how much my protein is moving, how much it is showing variation in its positions. So eigenvectors will tell you directions, at what direction, in which direction it is showing the change. So first eigenvector often represents the largest motion. So first eigenvector will capture the largest motion, which could be related to global changes like folding or large conformational shifts, whereas second and third eigenvectors, it capture the smaller changes. So it is also possible that my protein, it is only moving a little, whereas in other way it can show a bigger change. So first eigenvector, it calculates the bigger change, that means a global motion which is bringing about the change in the entire protein, whereas a little bit at the residue or the smaller peptide levels, they comes under second and third eigenvectors, that is local changes. So such as localized loop rearrangements or domain shifts, so they are captured by the second and third eigenvectors. If we talk about eigenvalues, so they quantify the amount of variance. Eigenvectors tell you the direction in which change is happening. Eigenvalue tells you how much change is happening, so which is associated with each eigenvector. So larger eigenvalues corresponds to the direction of motion that account for significant changes. So as we have seen that first eigenvector is capturing what is the most means what are the means larger motions which are present, whereas eigenvalue will tell you how much is that motion, what is how much impact or what is the amount of that variation. So in that way eigenvalue tells you that if the eigenvector is large, then it will also have the larger eigenvalues. So it will tell you that if it has the larger eigenvalue, that means that is a significant change in the structure. If eigenvalue is small, that means it is a minor movement, the amount is less. So we do not majorly focus on these minor movements because they are naturally taking place, so they are not bringing about any bigger change in the protein conformation. So for example, if the first eigenvalue is much larger than the other, then it suggests that most of the protein's motion is driven by the one major conformational change, such as folding or domain motion. So in this case, this is principal component 1 towards x and principal component 2. So principal components, it identifies that how much, in which direction the most variation is observed. So this represents the open state and this represents the closed state. So this is a dimensionality reduction method, principal component analysis. It tends to reduce the dimensions instead of considering multiple factors. So we only consider these two factors, PC1 and PC2, principal component 1, so direction towards which the major variation is observed, and PC2 is the second direction in which the major variation is observed. So you can see how the open and closed conformations, they are making different clusters. So you can figure out that if my principal component 1 and 2 value lies here, then this is the closed protein conformation. Yeah, so this is how the different protein states are represented at the structural level. So in this case, you compare multiple features and then find out that whether my protein, it is in closed state, open state, or what is the current conformation. And it also tells you about the protein dynamics, that how it is changing. Structure modes are provided after performing PCA. Then cavities dynamics, so we can also see that how my cavities are changing. So PCA will tell you about different cavity modes, that what are the different orientation your cavity is taking. So cavity is like a binding site, you can say, where the ligand binds. So there are certain cavities in the protein structure. So those changes you can study, and with the PCA you will be able to means there are different features. So instead of focusing on multiple features and then giving some label to a protein structure at particular time point, you perform PCA so that you can cluster them, that in which conformation they are currently present in. So that's why there are different modes which are possible. So this is about PCA. Then next is the normal mode analysis, which can also be performed. So this is a fast and simple method which is used to calculate the large scale motions in biomolecules. So for larger motions or large scale motions, you go for the NMA. This is a typical application and it is generally used to predict the functional motions in the proteins. And it also calculates the RM-CIP, in which the overlaps between the protein conformation space during successive time scale of the MD simulation can be measured. So in this case, we are with time period, we are seeing that how the conformation is changing. So NMA takes into consideration large scale motions in which the entire protein, how it is changing. Not just a smaller part is considered, but the entire protein, larger motions are considered, such as foldings or different things. Because in PCA we were focusing on the domain, we will also be focusing on the domain level orientation, so that is a local movements. But in NMA it is generally at the level of large scale motions. And in NMA, Hessian matrix is used in place of the covariance matrix as was used in the PCA. So in PCA we use this covariance matrix, whereas in NMA we use Hessian matrix. And what is the significance? So it helps us to understand the flexibility, and it identifies the low frequency vibrational modes. So because there are different atoms and residues which are present, so bond stretching, bond vibrations, they are taking place. So in this way we can capture those low frequency vibration modes which represents the large scale motions. So sometimes small vibrations can bring about the bigger change. So in NMA we can understand that change, such as domain movements or loop flexibility, which are key to protein function. So smaller changes brings about the larger effect that can be studied with NMA. Complementing MD, so NMA provide insights into the global flexibility of proteins, which complements the MD simulations. So after MD simulations, we can perform this NMA by focusing on essential large-scale motions useful for understanding the protein behavior or on the longer time scales. And it is also helpful in predicting the conformational transitions, and it helps to predict the structural changes and flexible regions which are involved in the folding, ligand binding or allosteric regulations, which are essential for the protein function. And energy landscape can also be explored, and it is also computationally efficient because it studies large scale motions, so without extensive computational resources. Because when you were performing MD simulations, then in MD simulations we perform it at the level of nanoscales, femtoseconds, picoseconds. So in NMA, because we are focusing on the global motions, large scale motions, so we are not that much restricted towards the femtosecond scale. We capture it at the level of, for example, microseconds, milliseconds. So time scale is increased. So if bigger time scale is considered, then that will take less time instead of considering a small time scale. So that's why it is computationally efficient, because it is focusing on the large scale motions. Yeah, and this after performing the NMA analysis in this paper, they were able to identify that this is the wild type, and this is the when there is a mutant at the 564 position. So glutamine, that is Q, is replaced with histidine, that is H. So by replacing it with histidine, so as we know histidine is positively charged, so if you replace Q with H, then it will bring about the change in the conformation. Global change is brought about by this change. So this is the wild type structure, and this one is the mutant structure. So you can see in both of them there is the bending. So this region is the hinge region, and if we consider this as a central axis, then we can see this particular means the region, it is bringing about the bending and deformation. So this is the hinge region, and there is this particular point, there is deformation and there is bending. So across this region there is bending and deformation. So with the mutant there is change in the conformation or orientation of the entire protein. So this can be tracked by studying the normal mode. And this is the workflow of NMA. So how we proceed with this NMA. So firstly we obtain the protein structure, because we have seen that NMA is complementary to MD. So in NMA we obtain a PDB file. We get the structure from experimental data or predictions such as AlphaFold. Then we check for missing atoms, which we did in MD simulation as well. We replace those missing atoms, we add those missing atoms by using different tools, by using PyMol, Modeler, or we can also use WinCoot. Then we undergo with energy minimization. We minimize that structure, and we can perform energy minimization to remove steric clashes. And for this we can use different force field, Gromax, Amber, and all those things are kept same. Then calculate the Hessian matrix, because in NMA we use this Hessian matrix. So this is very important, which was not used in that MD simulation. So Hessian matrix helps to compute the protein vibrational modes. And then we use NMA software, which is Brody, to calculate the Hessian matrix, and we analyze the low frequency modes. So low frequency modes means the collective motions which are related to function. So we perform the eigenvalue decomposition and analyze the significant modes to interpret the protein dynamics. So we have seen that eigenvectors and eigenvalues, so they tells you in which direction major changes are appearing and by what value that changes are. So by performing that decomposition we can identify that which of my modes or in which direction more changes are seen. So this is the flow chart of the NMA workflow, and this is the mode visualization. So this one is the mesophilic protein, whereas this is the thermophilic protein. So in NMA we can visualize the mode, and it shows a vector field representation of the modes. That is, vectors are used to show the direction and relative amplitude of the displacements undergone by different parts of the proteins. So these are different position vectors. This tells you in which direction major changes are brought. So we can compare two structures. So in this case they have compared mesophilic and thermophilic proteins to show you that how the conformation changes of two different proteins and towards which direction it is changing. So all these analyses can be performed with NMA. Yeah, and this is for your understanding. So I have given a table in which I have compared NMA with PCA. So if you are interested, you can go through the slide. I will not go into detail. And in this also there was a paper in which they have mentioned that we can use both NMA and PCA and we can get different information such as fluctuations, mode clustering, and by using PCA and NMA we can see that which directions are majorly showing change. So ultimately we can study that with time how my protein is changing conformation and either those conformations changes are functionally relevant or not. Yeah, and we can use different tools to visualize these results, such as VMD can be used, in which we can load the MD trajectories and we can compare it. So these are some of the steps which you can follow. You can upload this GRO file, which is the coordinate file in VMD. You can load the data. These are the trajectory files, and now trajectory will be displayed for analysis. So by following the steps, you can understand, you can compare the structures, that how the molecular dynamic simulation is bringing about the protein conformational changes. Yeah, and this is another paper in which stepwise how the MD simulation are performed and how the results are analyzed is mentioned. So if you are interested, then you can study this paper. So because this was quite easy and different commands are also mentioned. And then there was another paper which was talking about this is the review which talks about advances in MD simulations and enhanced sampling methods that study the protein systems. So in this is just an overview. This shows you that what are different things that you can study with MD simulation, such as binding site prediction, protein dynamics, binding affinity. So if you are interested, then you can go through this paper. This will tell you that what is the application of MD simulations in the real world. So now we'll quickly practice few MCQs. So the first question which is there is when we convert the PDB file to .gro file, then which file will not generate? So these are the options: .gro, topol.top, posray.itp and .xtc. Okay, let's see the right answer. So the right answer is option D, which is .xtc. So when we talk about GRO file, so it is generated during the conversion because it is a GROMAX format file which is used to store the atomic coordinates along with the box information. If we talk about topol.top, then this is a topology file which includes bonded parameters, force field information. So means it is not generated when we convert PDB to GRO file. Then posray.itp, so this is the file which contains the restraints, position restraints. So posre, that means position restraints for atoms, whereas .xtc, it is a trajectory file for storing the simulation data over time. So it is not generated during the conversion from .pdb to GRO. So it's produced during the simulation. That's why this one is the right answer, because this file will not generate when we convert PDB to GRO file. Now next question is, in which time frame duration the protein folding of a protein occur? Option A, millisecond to second. Option B, nanosecond to microsecond. Option C, picosecond to nanoseconds. And option D is microsecond to milliseconds. Okay, now let's see the right answer. So the right answer is milliseconds to seconds. So this is the time scale where the protein folding takes place. So if you want to analyze that how the protein is getting folded, then MD simulations need to be performed at this time scale. Then only we will be able to capture the protein folding. Because if you although if you perform at the micro level, then also you will be able to observe it, but it will take a lot of time because these changes are happening at the millisecond and second scale. So that's why we check that what vibration or what changes we want to study. So on the basis of that, we decide what should be my MD simulation run time. So next question is, which of the following is not bonding energy term? Option A, bond stretching. Option B, angle bending. Option C, rotate along the bond. And option D is hydrogen bond. So you have to tell which one is not the bonding energy term. option D. Yeah, so this one is the right, because hydrogen bonding is non-covalent interaction and it is not a bonding term, whereas all these three are the bonding energy terms. Next question is, which physical property is constant in microcanonical and canonical ensembles? So option A is number of atoms and volume. Option B is number of atoms and temperature. Option C is temperature and volume. And option D is energy and temperature. Okay, now let's see. So the right answer is number of atoms and volume. So when we were talking about the NVT, we were saying that it is also known as canonical ensembles, so in which volume, temperature and number of atoms were fixed. But now you will see that why option B is not correct, because in this case they are also including microcanonical ensembles. So microcanonical ensembles also considers the atom and volume to be constant. However, temperature is not fixed in the microcanonical, that's why option A is correct. Next question is, Gromax uses which force fields? Option A amber, option B charm, option C chromos, and option D is all of the above. Can you write in the chat box, because your audio is not recorded? Okay, so you mentioned option D, so you are right, all the above, because all these force fields are present in Gromax. Now next question is, which Gromox tool or command converts the proteins .pdb file to .gro file? So .gro is the Gromax format and .pdb is the protein data bank format. Option A is pdb to gmx, option B is editconf, option C is genion, and option D is grom. Okay, so you are saying option A. Yeah, that's right. So option A is right, which is pdb to gmx. It converts .pdb file to .gro file, which is Gromax version. And then these are some other commands, editconf, then genion and grom. Okay. Now next question is, in normal mode analysis, which matrix is used for calculation? Option A covariance matrix, option B Hessian matrix, option C diagonal matrix, and option D is Jacobian matrix. So this also we discussed when we were talking about the NMA. Okay, now let's see. So the right answer is option B, which is the Hessian matrix. So when we were talking about NMA, then we studied that there is the involvement of Hessian matrix, whereas in PCA we were talking about the covariance matrix. Okay, so now next question is, what are the conditions for minimum, maximum and saddle stationary points in the potential energy space respectively? So these are some of the equations. You have to tell that which one is the minimum condition, which one is the maximum, and which one is the saddle stationary point. So this tells you about the derivation with respect to potential energy. Okay, so it might sound a little mathematical, so let's see the right answer. So the right answer is option B. This is right, in which in this case this derivation is greater than 0. Second derivative of potential energy towards x and y axis with respect to derivation towards the x square. So the energy value is this derivative is greater than 0, and then in this case the derivative is less than 0 in the case of maximum, in minimum greater than 0, and in saddle point towards this x square it is greater than 0 and towards y square it is less than 0. So now in case of minimum, all the eigenvalues, they are positive, that means stable and energy is lowest. So there are positive eigenvalues which can be seen here, so value is greater than 0. Whereas in case of maximum, all the eigenvalues, they are negative, so that's why value is less than 0. So this is calculating the eigenvalues, which means it is the unstable state and energy is highest. Whereas in saddle point, it is a mixed positive and negative eigenvalues. So in this case, one of the eigenvalue is positive, that is greater than 0, whereas other one is less than 0. So in this way option B was right, and if you are interested, then I have also mentioned in detail what each point means. So the conditions for minimum, maximum and saddle points, they are determined by the behavior of second derivative which is obtained from Hessian matrix of the potential energy function. So minimum means local minimum. So conditions means all the second derivatives of the potential energy with respect to atomic coordinates, if they are positive, then that means it is in the local minima state. Maximum means all the second derivatives of potential energy, they are negative. Saddle point that we have discussed, that if the second derivatives are mixed, some are positive, some are negative. So in Hessian matrix it has both positive and negative eigenvalues, which represents a transition state where the potential energy increases in some directions whereas it decreases in other directions. So this is often the peak of energy barrier in the reaction pathway. Now let's move to our next question. So the topology file may specify option A bonds, 2 atoms connected, option B dihedrals, 3 atoms connected, option C angles, 4 atoms connected, and option D is angles where 5 atoms are connected. So are you aware of this question? Option A. Okay, let's see. Yeah, so you're right, option A is correct. Bonds means 2 atoms are connected, because we have seen when we study dihedrals, then it must require 4 atoms. With 3 atoms we cannot make dihedrals, and for angles we require 3 atoms. So that's why the right answer is option A. Now our last question for the session is, with respect to which structure of the trajectory of MD simulation root mean square deviation is calculated? Option A is final structure, option B is initial structure, option C is the clustered center structure, and option D is the random structure. So what do you think? With respect to what do we calculate the RMSD? Are you saying C or B? Okay, so the right answer is option B, that is initial structure, because we have seen that to calculate the root mean square deviation, we have initial structure, then we have a final structure obtained after MD simulations. So we compare these structures to find out how much my final structure varies from my initial structure. Yeah, so this was the last question for today's session. So if you have any doubts, then you can ask, otherwise I'll end the session. Okay, thank you. So now I'll end the session. See you in the next session.